My name is Uyen Chu. I am the Senior Research Associate, a team lead at Corteva Agriscience. Today, I'm going to walk you through the DOE experiment. The DOE experiment walkthrough include the problem statement, the benefit of DOE methodology, the DOE execution roadmap. You have heard Randy talk about the principle of why we use the DOE to solve a tissue culture problem. Now I will walk you through an example of the DOE, how to set it up, and what we get from the data that collects from the experiment. First, I have a scenario. A tissue culture lab decides to explore a new process. The goal is to maximize the number of root plants to send to the greenhouse. There are many factors might contribute into tissue culture. Before running the experiment, a group of experts gathered to talk about the factor we want to test in the experiment. If anyone works in tissue culture, we know that there are many factors that could impact to the result: environment conditions, media components, or many more. From a bunch of factors, the team narrowed down to 8 factors. If we want to use traditional methods, 1 factor at a time, OFAT with 8 factors, 2 levels, 256 experiments need to run. Label intensive also cannot detect the interaction effect. Besides what factor to test, what to measure is also very important. We want to measure what relevant to the experiment, to the project. Before I walk everyone step by step how to set up the DOE experiment, I want to say again the benefits of the DOE methodology. The DOE methodology should be picked to when we have multiple factors and we want to see the interaction between these factors with fewer tests. Here is the DOE roadmap. Randy have walked us through step 1 to 5. I'm going to show step 6 to 8. Step 6, we will design the experiment by using fractional factorial option in the design expert. This will be the software that I will use for my talk. To remind everyone, we want to screen 8 factors to identify the main effect, if any, between these factors. Then step 7, we'll run the experiment, collect data, analyze data, then narrow down the list to the factor to a few. Finally, step 8, we will run the optimization and confirm if we meet the objective goal. There is a few information, the term that we need to know when we use the software to design the experiment. First, this table generates by the design expert. You can find it with other software as well. This lists different fractional options. On the y-axis, that shows the number of runs, which is the number of treatment. The x-axis shows the number of factors. White area is the full factorial, 2 to the A power or 256 experiment. However, we should need to screen the A factor for those that have the main effect, and for that, the resolution 4 design is sufficient, the yellow box. For screening experiment, we can reduce the experiment to either 5 or 4. In this case, it's 2 to the A minus 4 or 2 to A minus 3. And we choose 4 and 2 to 4 is 16 experiment. In this case, the yellow area is where we need to pick. The green area is not chosen because number of runs still plenty for the screening purpose. And we want to avoid the red area. The resolution 3 is too simple and we cannot get enough information to discuss the main effect. When we choose a fractional factorial, you eliminate some of the combination of the factors. As a consequence, alias or confounding occurs when using fractional, factorial, and one combination represents several other. In particular, these are the aliases in resolution 4, design that is being used to demonstrate. Alias is the confounding interaction between factors. This is baked into the experimental design by the software. This is the list of the factors we are going to use for the demonstration. The first column is the code for each of the factors for easy when we design the experiment. So the second column is the name of the factors. And this one is the unit for each of the factor. And low and high is the range. So the range for each of the factor need to chosen wisely, wise as we can, but reasonable. High and low depend on the system. Use whatever the historical data or literature or anything that can give an estimate of the range. We don't want it is too narrow that we might miss the optimal setting, or we don't want it too big, so we might miss the result. And also we note here that the factor can be two choice, either numeric or categoric. In this case, we have three factors that is categoric, is the carbon source or gelling agent or vessel venting. So for vessel venting, either we can do non-vented or vented. So we can combine these two type of the factor. Next, let's see how to design experiment with the design expert software. And to remind, we have what we have. We have a factor and factor that have two levels, low and high. And we use the fractional factorial resolution 4 design with 16 run. Which is 16 treatment for this experiment. And this is the first page of the design expert. When you open the design expert, you will choose the new design option, and then you just follow step by step. In the software, it's very clear to tell us how to create the experiment. But I want to mention here that the first step for screening is we use the randomized option with the regular 2 level for the screening experiment. And then we just follow the instruction in the software. And then at the end, we will generate this table. We can see that we have 16 run. That is the number of treatment we decide we would do this time to find the main effect from this 8 factor. And factor 1 to factor 8. And we see here they include both the real name and also the code for each of the factors from A to H. And the last column is the response. We can add more if we want, but in this case, I only have one response I want to collect is the number rooted plant. And in here, you can see the number for each factor. For example, factor 1, you see 1 and -1. When you design the experiment in the software, there is the place you can either enter the real value of each factor, low and high, or you can just leave it -1 is the low level and 1 is the high level for simplify. So in this case, I just leave it. And then you will go on and do the experiment and then collect the data. I want to mention again, and what to measure is very important, similar to the list of the vector. So we want to measure what need to generate meaningful result and no need to measure everything possible. And also we can take economic into the consideration. For example, sometimes we have to consider between geotin and BAP. So geotin might show the best result, but BAP, the result might be close to the best. But BAP is a lot cheaper than geotin. Then sometimes we want to go with BAP option. So this table is just repeat the table earlier that I showed, but we have the last column field. That is the data that we collect from the data. And the objective goal for this experiment is 7 plant or higher. So some of the treatments show promising results. So the red number is showing either 7 or higher. And we will analyze the data using the Design Expert tool. The data come out from the experiment will be screening. The software does numerous quality check on the data, such as ANOVA and analysis for the outlier. I will walk through some of them through my talk. The first is check normality by normal plot and Buck-Cox plot. The first graph is the normal plot. So we expect all the data plot lie within the straight line. If not, the data might need to be transformed in order to normalize the data. And how do we know that if the data transform or not? The second graph is the Buck-Cox plot can tell us that too. So the recommend lambda in this case is 1, and it is the power to which you raise the data to make it normal. So in this case, the lambda is 1, and we do not need to transform the data. And if the model recommend to transform the data, we can use many functions such as square root, power to transform the data to normalize it. And the Design Expert is the nice tool that will tell you whether you need to transform or not. In this case, it's a recommend transform in none, so you don't have to transform the data. And I have been using Jump and Minitab. That's one. The Buck-Cox option is not available, so you have to use the normality plot graph to check whether you need to transform or not. So if the data is not lie within the straight line, then yes, you might have to check the data whether you need to transform or not. The first so after check the normality of the data, then you will, from the data diagnosis, the D5 versus run, that's the next one we want to check. This one, it's so from the first graph, we can see that run 15 in the red circle is the outlier. So we go back and investigate. After investigate, we realize that there is a mistake, just a typo when the data collected. And the graph 2 show the data after corrected. D5 is the differences in the fit test. And one thing we have to mindful that if we're not sure about what happened, when we will choose to the option to ignore the data point, then this data point would not include when we run the analysis with this data set. Again, the DOE software have series of tests to check the quality of the data. It makes sure that this data is high quality and we can trust the analysis and the result. So the next one is the last quality check we do is to check the R square value. In this case, the R square value is 0.9447 and predict R square is 0.8584 is in the range with adjust R square of 0.9170. So the difference between these is that less than 0.2. So it's all acceptable. Another step to check the data is by checking the ANOVA for p-value. From ANOVA table, the p-value less than 0.05 indicate model term as significant. So in this case, A, B, D, and the AC as significant model term. These are the main effects. Value greater than 0.1 indicate the model term are not significant. Next, we can visualize this data in Pareto plot. From this graph, we know that AD and AC is the main effect, significant since it exists a Bonferroni light. Each experiment will have different Bonferroni light because they will calculate based on the real data. Another exciting thing about the DOE methodology is the power of the DOE is to detect the interaction between factors, indicated by the line crossing in the third chart. The first graph, we can see low setting of A factor, we will get low rooted plant number, and high setting will get higher rooted plant. With C, the setting will not make the difference, but with the interaction, we might want to check the setting at the different setting for each of the factor. To remind us again, AC is the main effect, but we are not sure what it is because it could be the interaction between BE or DG or FH. The question we can answer is if there is an optimal point between two levels, low and high, because in the screening, we only test these two areas, either low or high. We don't know the answer in the between. So we will determine the optimal level when we run the optimization experiment that generate a response surface methods, and that I will talk next. To remind us again from the Pareto chart, A, B, and D were the main effects. Those factors will advance to the next step to run the optimization experiment. C is the carbon source, and it's not the significant impact to the result. We can use either sucrose or glucose. So in this case, I chose to use glucose. Then we will go back to use the design expert to design the optimization experiment. This time we will choose response surface option and with optimal option and follow the instruction in the design expert to generate the table similar with the screening experiment that we talked earlier. And with the response surface method, we can see the interaction between these three factors that we test. The first graph shows the D, which is the BAP at 0. We can see that at 50 micromole light intensity, potassium nitrate at 40 millimole, we will get the highest number root plant in the orange circle. And the second graph shows the same trend at 50 micromole light intensity, potassium nitrate at 40 millimole. But when we increase the light intensity, the number of rooted plant decrease, which is in the red circle. And for this one, we check with the AB, and the changing factor is the D factor. We can do the similar with AC or BC when other changing its value. Then we will see the we will find the best option to run the confirmation experiment to confirm whether we meet the objective goal or not. And to summarize my talk, for the DOE experiment, take-home message is we, using the DOE methodology, we can save time and labor by testing multiple variables at once. And we need to choose the factor and the range carefully. And we need to choose what to measure for meaningful result. From the DOE, we can create a rich data set, capture interaction between factors. And DOE software is the necessary in order to design and analyze the experiment. When we run the experiment, we need to be extra caution due to the complexity of the design. And we need to choose appropriate experiment design for the design outcome, such as screening design for identify factor with major effect. And then we narrow down to a few factors, and that time we will use the response surface method option to find the optimal setting to confirm the result. With that, I will conclude my talk with our contact information. Please reach out to us for any question related to the DOE methodology. We were happy to take any question or further discuss about this topic. Thank you.